Hello, welcome back, friends. This is Life Science, and this is your human physiology. And in this human physiology lecture, we will focus on the hemoglobin dissociation curve. So, what is hemoglobin dissociation curve? How to define the oxyhemoglobin dissociation curve? This curve shows the how changes in the partial pressure of oxygen influence the oxygen binding to hemoglobin and oxygen dissociation from the hemoglobin. So first of all, why we plot this type of graph? For better understanding of the dissociation curve, and also you call it as a association. Remember, here is association and dissociation, both can take place in this graph. If we talk about let me change the color. So if we talk about the y-axis, this is the y-axis and this is the x-axis. Remember, when you want to read any graph, you have to remember y-axis and axis. What values we put on the y and what values we put on the x-axis. So y-axis we have put on the percent saturation of hemoglobin with the oxygen, and on the x-axis we have plotted the pressure of oxygen in mmHg. So percent saturation of hemoglobin as a function of pressure of oxygen. So pressure of oxygen, so percent saturation. If we talk about percent saturation, this value range from 0 to 100. This value range from 0 to 100. And if we talk about pressure of oxygen, this value is range from 0 to 100 mmHg mercury. 0 to mercury. So suggesting that as pressure of oxygen is increased, Hg% with hemoglobin saturation exhibit a sigmoidal curve. So this is the sigmoidal curve. This whole is the sigmoidal curve, suggesting that there is a cooperativity with respect to oxygen binding to hemoglobin. Now, what is cooperativity? Let's understand this. There is a 4-heme group of hemoglobin. So this is the 4-heme group of this is the heme group of hemoglobin. This is the site where the oxygen molecule can bind with the hemoglobin. So first of all, if the one molecule of oxygen can bind this region, so it will facilitate the another heme group of hemoglobin. This in turn shows the cooperativity. That means one molecule can facilitate the another molecule. If we talk about pressure of oxygen at 0, remember we have started with the 0 to 100% saturation. 0 to 100% is saturation. So we will discuss about. So we will discuss about. So we will discuss about on the at so we will discuss about at when pressure of oxygen will be 0 mm mercury. Now in this time what happened? And at pressure of oxygen 100 mm Hg. This time what happened? And at pressure of oxygen 40 mm Hg. So 40 mm Hg, 40 mm Hg, 40 mm Hg. In this time what happened? When will the 40 mm Hg, when the 100 mm Hg? This graph shows all the activities in a single graph. So at pressure of oxygen in 100 mm Hg, that means this shows no oxygen molecule bind with heme group, no oxygen bind with heme group. So in this time no cooperativity phenomenon can observe. When we talk about the at pressure of oxygen 100 mm Hg, in this time all the heme group get saturated with the oxygen molecule. All 4 heme group get saturated with the oxygen molecule. This is the arterial blood. This blood, what we can say, arterial blood. And this is the venous blood, or in other word we can say deoxygenated blood. This is the oxygenated blood, or this is the oxygenated blood. So in this time when we talk about the 40 mm Hg, when we talk about the 40 mm Hg, approximately 75% of the binding sites are occupied by the O2. And remember when we talk about the 100 mm mercury, in this time 100% saturation, but not 100. Remember, 98% saturation can take place. So all the binding sites are occupied in the 100% saturation, and at the pressure of oxygen 40 mm Hg, there will be the 75% saturation. This show the 75% saturation. This shows the 75% saturation. So if it is often used to refer the partial pressure of oxygen. So partial pressure can decrease and can increase. Both phenomena we can observe in the single diagram. So this referred to as the CO2, as the O2 partial pressure increase or decrease. This phenomenon can change the affinity for oxygen. If we talk about P50, now what is P50? Before going to graph, we have knowledge about the P50. This is the value at which 50% of oxygen molecule gets saturated with the hemoglobin molecule. In this time, pressure of oxygen will be approximate 28 mm Hg in normal, or you can say healthy individual, or in normal, or you can say healthy individual. In normal, or you can say healthy individual. In normal, or you can say, so there are some factors which decides the pressure of oxygen, where the pressure of oxygen get increased, where the pressure of oxygen get will decrease, that this is the get decrease. So decrease, decrease. So there we will have some factors. So there we will have some factors which will affect the pressure of oxygen, and in turn to percent saturation of oxygen molecule with hemoglobin. So there are several factors. These several factors, there are some several factors. So what are the several factors? First one is the temperature. First one is the temperature. First one is the temperature. Now what happened when temperature increase or decrease? We'll see in the when the graph dissociate. So temperature. First factor is the temperature. Second factor will be the pH. pH is also influence the pressure of oxygen and percent saturation of oxygen with hemoglobin. And third one is the partial pressure. Why we call it as a partial pressure? Remember, there is a gradient, concentration gradient of oxygen molecule. Oxygen molecule, not the oxygen molecule. Here we will have the pressure of carbon dioxide molecule. This will also affect the pressure of oxygen. This will also affect the pressure of oxygen. This is the carbon dioxide. Okay. So these are the some factors which will affect the temperature, pH, which will affect the affinity of oxygen for the hemoglobin. This is the temperature, pH, and partial pressure. So if we talk about blood, this is the, suppose we have 100 ml of blood. This is the 100 ml of blood. So in 100 ml of blood, how can calculate the oxygen concentration, or how much oxygen is present in this 100 ml of blood? How much oxygen is present in this 100 ml of blood? How this? So we have at pressure of oxygen 100 mm mercury. Remember, we can talk about the healthy person. So in this time 20 ml, this is the percent value. This is the whole blood is the 100%. This is only 100 ml हम ले रहे हैं, 100 ml, तो 20% ml. If we talk about at pressure of oxygen, this is the more and more advanced values for better understanding to the oxygen hemoglobin dissociation curve. This is the conceptual part. So this is the conceptual part. So in this time 40 mm Hg. Now in this time what happened? 15 ml blood, 15 ml of oxygen in whole blood per 100 ml. This is the venous blood. And this is the arterial blood. we have some physiology concept that will require for better understanding. This is the arterial, this is the venous blood. At pressure of oxygen, at pressure of oxygen 0 mm Hg, इसकी भी बात कर लेते हैं 0 mm Hg. So in this time 0 ml% saturation of oxygen molecule with heme group of hemoglobin, with heme group of hemoglobin. So note, I have some notification point that will be remembered, as it is important to see that while venous blood is often referred to as the deoxygenated blood, it will still contain a significant amount of O2, that is the 75% of O2. This is the arterial blood. Remember, in arterial blood we have 100% saturation, but in venous blood we have 75% saturation. Only 25% oxygen will unloaded. Only 25% oxygen will unloaded. Only 25% of oxygen will unloaded. So arterial blood contains 20 ml of O2 for every 100 ml of whole blood. For whole blood, meaning that nearly 100% of hemoglobin O2 binding sites are occupied by the O2. O2 binding sites are occupied by the oxygen molecule. So we will switch on the initial diagram of the oxygen hemoglobin dissociation curve. In this diagram, what we want to set, I can set that this is the simple representation of the hemoglobin dissociation curve. How the hemoglobin can dissociate with the oxygen molecule and how the hemoglobin can bind with the oxygen. So we have some concept regarding this. So we have this is the alveolar, or this is the blood capillary. So in the blood capillary, this is very thin, even one RBC, its diameter is very thin. Even one RBC can move at a time. Even one RBC can move at a time. Only one RBC can move at a time. Only one RBC can move at a time. So in this time, what happened? This is the alveoli. So pressure of oxygen in alveoli is the 104 mm Hg, and the pressure of CO2 is the 40 mm Hg. So, and this is the deoxygenated blood, this is the oxygenated blood. So this is the RBC, and this is the RBC, and this RBC have the CO2 in more concentration and O2 in. So this alveoli, there is the phenomenon of diffusion. This is show the diffusion. So via the diffusion, oxygen molecule can bind with the RBC, and in contrast, in response to CO2 will be removed out from the RBC. So if we talk about, this is the alveoli. Remember, I can say in alveoli pressure of oxygen will be 104. Here we have written the 100 mm Hg. This value vary from book to book, but this is the standardized value. And if we talk about pressure of carbon dioxide, this is the 40 mm Hg, 40 mm Hg, or you can say mercury. So in this time there will be the oxygen is the fully saturated, or hemoglobin is fully saturated with the oxygen molecule. In this time we have talking about the pressure of oxygen at 100 mmHg. This is the value for the alveoli. In this alveoli, this is the value for the alveoli. In contrast to RBC, there is the presence of tissues. So this is the tissue. This value is for the tissue. This value is for the, remember there is 75% saturation of oxygen molecule. 75% saturation of the oxygen molecule. So this is the oxygenated and this is the deoxygenated. Still there is the 75% saturation or 75% of arterial blood is present. So now in this time, if we talk about the human physiological resting range, so this range from the 40 mmHg to 100 mm mercury. This will range from this is the normal, or you can say healthy person. This is the normal, or you can say healthy person. So if we talk about at P50, what P50 denotes, I will tell you. P50 denotes that is this is the percent saturation of oxygen, where the 50% oxygen molecule can saturated with the hemoglobin. So at this point, pressure of oxygen will be approximate 28 mmHg. This is the normal range. So how we can define the 28 mmHg? So in this time, 50% oxygen molecule unloaded. 50% oxygen molecule will unloaded at that time. So here, when we studied about the oxygen dissociation curve, we have three values which will be the indication or for the key points for better understanding of the graph. This is the you have remembered the pressure of oxygen 28 mmHg. What happened? Second, pressure of oxygen when 40 mm. In this time, what happened? This is the resting and this is the vigorous exercise. When we exercised, this value goes up to the 28 mmHg and pressure of oxygen at 100 mmHg. These are the values which will cover the whole oxyhemoglobin dissociation curve to better understanding. When the oxygen, this is the 100% saturation, so this will show the dissociation. There is the presence of tissue will require the oxygen molecule because they have lots of energy generation. So this is continuously demand for the oxygen molecule. So this is the oxygen dissociation. There is the dissociation of oxygen, and this whole process is taken place in the RBC. This whole process is taken in the RBC. The RBC move from blood capillaries. This is the blood capillary. And exchange occur in this manner. So when we talk about the oxyhemoglobin dissociation curve, there will be three values which will remember for better understanding. This is the value for resting physiology. We have not working at all. I have only rest, and this time 40 mm to 100 mm of Hg oxygen 40 mm pressure of oxygen get occur. And this time when we vigorous exercised, in this time we have up to the P50 level 28 mm. Okay, so P50 is the, I have told you P50. So in normal condition P50 is 28 mm mercury, and we will 28 mm, and when the oxygen affinity decrease, so this will lead to the 30 mm Hg, means P50 value get increased and when the affinity oxygen affinity for Hb increased, this value is reached up to the 30 to 50 mmHg, 30 to 50 mmHg. This phenomenon observed in fetal hemoglobin. This phenomenon is observed in the fetal hemoglobin. This phenomenon is observed in the fetal hemoglobin. Why we repeat more and more? Because you have hearing and hearing more for better and better listening. So if we talk about this graph, what this graph shows, actually examiners question sets from this part. How? So this is the normal, I have written this is the resting physiological, this is the resting physiological. What happened when this graph tilted towards this or tilted toward this? Tilted toward this. What happened? When the graph shift right shift graph, this phenomenon is given by the Bohr. Bohr is a scientist. Bohr's, इसको कहेंगे हम Bohr's effect. This will be the Bohr's, and this is the Halden effect. This is the Halden effect. So this is the Bohr's effect. This shows the dissociation. This shows the dissociation, and where the dissociation, which side, which will show in the tissue site, and Halden effect, where this is the alveolar site. Remember, in alveolar site, pressure of oxygen 104 mmHg, pressure of CO2 40 mmHg, and in contrast to Bohr's effect, there is the pressure of oxygen getting decreased up to the 40 mmHg, and pressure of CO2 will increase 45 mmHg. So what factors can determine where or whether the normal dissociation curve shifted or tilted towards the right or tilted toward the left? There will be some factors. First of all, we will focusing on the dissociation. Remember, this is the dissociation part. So in this part, decreased affinity for O2, and this part, oxygen molecule have saturated up to the 75%. So what will happen at this time? This is the tissue site. RBC can move up to the tissue. This is the tissue site. When we talk about the resting phase, this is the 28 mmHg. This is the 20 mmHg. When the 50% oxygen molecule get saturated, this is the normal. And when this normal, this is the affinity, pressure of oxygen, and when the pressure of oxygen getting 30 to 35 mmHg, this will show the decreased affinity for O2 of hemoglobin. And when the hemoglobin have affinity 30 to 50 mm mercury, this will show the left hand shift of graph. So in the right shift graph, pressure of CO2 will increase, H plus ion increase. So as the H plus ion increase, pH will be low. 23123 BPG also increase in this case. And what can happen when 2,3 BPG increased at that time? This is the competitive inhibitor of, this is the competitive inhibitor of oxygen. So where the oxygen get bound, at that place 2,3 BPG can bound with hemoglobin and the temperature get increased. This phenomenon, when these all factors or these phenomena occurred, in this time normal curve shifted toward the right. Normal curve shifted toward the right. Normal curve shifted toward the right time, toward the right. So what happened when the curve shifted towards the left? If we talk about left shift, in this time increased affinity for O2. This happened at what time? When you have fetal hemoglobin. This time fetal hemoglobin. At this time, pressure of carbon dioxide will decrease, decrease. As a result, pH will be high pH. As a result, pH will be high. 2,3 BPG concentration also decrease, and temperature. This will show the hemoglobin of fetal hemoglobin. This will show the fetal hemoglobin. So questions come from this part only. So to better understanding from this lecture, we have memorized the pressure of oxygens at multiple, or you can say tissue at rest and alveoli sites. Both sides are memorized for the better understanding and for the better explanation. Thank you.